Welcome to Let's Crack NEET UG English. I'm your biology teacher, Dr. Neela Bakore, and we will be continuing with cell cycle. But before we get started, let us have the basic checks. If the audio is okay, if you are facing any problem, just let me know. I guess I'm audible properly. Audio is clear. That is Aarti. That's very good. Thank you. And as I said, we will be starting with the same thing that we did in the previous classes, and that was the cell cycle. Now, in cell cycle, we have done pretty much everything, and we started with the cell division part, that is the division. And we discussed mitotic division in detail. So now we'll continue with the same thing. And okay, so let's get started with our topic for today. We will be talking about significance of mitosis. We will also talk about types of mitosis, and there is a difference between mitotic division, which takes place in plant and animal cell. And then we will introduce the next special type of division, which is meiosis. And we will try to have an overview, plus we will start discussing the first part or the first phase. This first phase, that is Prophase I, it is very long, it is a complicated process, and it takes time for the students normally to understand it. So what we have planned for today is I'm going to start Prophase I, and in the next lecture we will revise Prophase I and then continue with the other stages. Alright? We are on Let's Crack NEET UG English channel, and this is about me. I'm your biology teacher. I take botany and zoology both for you, and we are using Unacademy platform. All these things I have told you, and don't forget to install the app from your app store, and that is Unacademy Learning app. And once you get this app, you know the basic things which you will be able to avail. There would be live classes, live tests, and you would have unlimited access to live lectures of many renowned faculty members who specialize in this particular stream, that is NEET UG. So you can get the live lectures of all these faculty members. I have talked about these plans earlier also, and I'm sure by now you have decided which plan you want to go for. And just decide the plan and start using this code for getting that plan, so that you get the 10% discount. So this is some basic information if this subscription plan is to be taken, and there is a wide range of possibilities that you can go for, a short duration plan to a very long duration plan, keeping in mind your preparation time. All right, let us come to the topic now. We are talking about mitosis. If I have to give you a recap of what we have done so far, we did cell cycle, we are still doing cell cycle, and we talked about all these changes which take place in G1, S, G2, and then was M phase. In M phase, that is multiplication phase. In multiplication phase, we have talked of two types of divisions. We talked of amitosis, we talked of mitosis. Okay, so we are still in the cell cycle, but we are talking about the cell division part. And because we have done this mitosis in detail, we have to talk about some more important things of this mitosis. I did give you a brief idea of the significance of mitosis in the previous lecture, but we will talk about this in little more detail now. Mitosis keeps the chromosome number constant. We know that chromosome number is species specific. For every species, the number of chromosome always remains same. So if there is a cell with say 20 chromosomes, when it divides by mitotic division, that chromosome number has to be maintained because it is species specific. This is what is meant by the first point under significance. Second is growth, repair and regeneration. This also we talked of. How is an organism formed? Say we are talking about ourselves. So a male gamete, a female gamete, they fuse, fertilization takes place and a zygote is formed. So this is the sperm, this is the egg. This brings, if we are talking about humans, then this sperm brings 23 chromosomes and the egg also brings 23 chromosomes, and in the zygote we have 46 chromosomes, which is the specific chromosome number for humans. Now from this zygote, a complete organism is going to develop, and all the cells of that organism, if we are talking of humans, the trillions or trillions of cells which make up our body, they all have 46 chromosomes. So which division is taking place here? Is mitosis. So it helps in growth, it helps in repair. Suppose there is a fracture in a bone. So the new cells which are formed, they are bone cells, they are diploid cells. So the repair is taking place. For the repair, the division which is there is again mitotic division. If some part is to be regenerated, then also there is the same type of division which is taking place. Now, if you're talking about lower organisms like amoeba, if you are able to recall, we said amoeba divides by amitosis as well as mitosis. Under unfavorable condition, because it has to survive those unfavorable conditions, amoeba quickly divides by amitosis, and that results into different types of cells, some with little more genetic material, some with little less genetic material, and once the favorable conditions return, amoeba divides by mitosis, and this is asexual reproduction. So for lower organism, mitosis is the only method of reproduction. Very few lower organisms divide or reproduce sexually. So these three important points come under significance. Now when we talk of mitosis, we have taken the general steps: prophase, metaphase, anaphase, telophase, and so on and so forth. There are some other types of mitotic divisions also where there is a slight difference, out of which two we have already talked of. For example, anastral. This we have discussed when we were talking about the steps of mitosis. Anastral is when no aster is formed. And what exactly is there in an aster? Aster has a centriole and the aster rays. So if we are talking about mitotic division in plant cell, where centrioles are absent, so there would be no aster formation, then the division will be known as anaster. It is mitosis, but because there is no aster formation, it will be called anastral. So where do we find anastral? In plant cells, as no centriole is present, and so no aster. So it is mitosis, but because there is no aster, we call it anastral. Now the second one, amphiastral, that means two asters. This is seen in animal cell. two asters are formed. An example is animal cells or the cells which have centriole. Now let us come to this third. We have not studied this. We just talked about the astral and anastral, which is which are the basic type most common in plants and animals. Now when we talk of intranuclear, it is mitotic division. That means it is equational, equal distribution of genetic material. Same number of chromosomes would be there in the daughter cells. But in this, there is no loss of nuclear membrane. If you remember what happens in prophase, the nuclear membrane, nucleolus, the membranes of the organelle, they start to dissociate. And the entire space inside a cell is now available for the chromosomes to move, right? That was the common type of mitosis that we talked of. Here, nuclear membrane does not dissociate. So what happens if this is a cell? The nucleus is going to remain intact, nuclear membrane is going to remain intact, and the spindle fibers, which otherwise in normal division what we saw was that the centrioles are at two poles and the spindle fibers are formed like this, right? Here the spindle fibers are formed inside the nucleus. So spindle fibers are formed, but everything happens inside the nucleus. What will happen? After anaphase, the chromosomes will move towards two ends of the nucleus, and then the nucleus will divide, and then the cell will divide. So chromosome number is going to divide, or rather I would say in both the nuclei, both the cells, the chromosome number is going to be the same as that of the parent cell. But instead of the entire space available, everything is going to take place only inside that nucleus. So no loss of nuclear membrane. Spindle fibers are formed inside the nucleus. So everything takes place, but instead of the entire cell, everything is going to take place inside the nucleus. Now, the next type, this is again very, very important. Free nuclear division. In this case, karyokinesis takes place, but it is not followed by cytokinesis. So if this is what is happening, what will happen inside a cell? Inside a cell, prophase will take place, that is nuclear membrane dissociates and all those things. Metaphase, chromosomes will come on the equatorial plane. Then anaphase, spindle fibers are going to pull the chromosomes apart. Telophase, nuclear membrane reassembles and you start seeing them. Nuclear division done, but no cytoplasmic division. So in this case, one nucleus is formed, the other nucleus is formed because karyokinesis is completed. But after that, cytoplasm doesn't divide. So now a cell which was having only one nucleus now has two nuclei. So by this method, the cells will become binucleate or multinucleate. So this division results in formation of bi or multinucleate cells. Which are these multinucleate cells? We can take example of endosperm cells, tapetum. If you have not heard of tapetum, it is a layer of nutritive tissue in the anther. When we talk of reproduction in flowering plant, at that time we'll talk about this. But for the time being, you can just remember that these are the cells where the multinucleate condition is found. And how is this multinucleate condition seen? Karyokinesis takes place but not followed by cytokinesis. So in a cell there would be 2 nuclei, then 4 and 8 and so on. So the number of nuclei is going to increase. This is free nuclear division. Dinomitosis is very similar to intranuclear mitotic division with a difference that here same thing, no loss of nuclear membrane. This is same, but inside the nucleus there is no spindle formation. In case of intranuclear, we said spindle fibers are formed inside the nucleus. Here no spindle fibers are formed. This is known as dinomitosis. So intranuclear dino, same, nuclear membrane remains intact, but in intranuclear spindle fibers would be formed, and in dinomitosis no spindle fibers. Free nuclear is karyokinesis normally takes place, but cytokinesis is missed or it does not take place. So these are some variation from the normal type of mitotic division. These two are normal, everything normal, but the one which takes place in plant is called anastral and the one which takes place in animal cells is called amphiastral. What is the difference between this same division, mitotic division, in plant cell and animal cell? We have understood the complete process and we can pick two differences. One, just now what we talked of, in plant it is anastral, no aster. In case of animal it is amphiastral. This is one difference. The second difference is seen in cytokinesis. In case of plant, the cytokinesis takes place by cell plate formation. If you are able to recall, what we discussed in the previous lecture was that if this is a plant cells have a rigid cell wall. And if there is a rigid structure, the plasma membrane cannot show folding. And that is why plant cells do not undergo endocytosis or exocytosis, like you have seen how amoeba engulfs food, phagocytosis. So the plasma membrane bends, it is flexible, it makes a cup-like structure and the food comes inside. This is infolding. Such processes are not possible in plant cell or any cell which is having a cell wall. So cytokinesis cannot take place by constriction of plasma membrane. So what happened here was telophase is complete. So these two nuclei are formed, nuclear membrane has reassembled. Then there were these phragmoplasts which come here. Phragmoplasts are fragments or pieces of ER and Golgi. Now these fragments are known as phragmoplasts, and this entire layer which is formed is called a cell plate. So what is cell plate made up of? Cell plate is made up of phragmoplasts. And then on either side the cellulosic layer will be deposited. So if these are the phragmoplasts, now the phragmoplasts themselves, they will start producing calcium and magnesium pectate. So this becomes the middle lamella. Middle lamella is formed, and now from both the sides, both the cells are going to deposit cellulose. So from this cell cytoplasm, there will be cellulose deposited on this side of the middle lamella, and from this cell, its cytoplasm is going to deposit cellulose. So this is cellulose of one side, and this is again cellulose of another side, and this whole structure is the cell wall. So this structure is going to be formed here and it grows towards the periphery. So it starts in the center, grows towards periphery. Whereas in case of animal cell, what happened was, in case of animal cell, the plasma membrane showed this kind of infolding. Because there is no cell wall outside it, the membrane is flexible, and these infolds, they get deeper and deeper, they get more deep, and finally the two cells will be formed, daughter cells. So in case of plant cell, it is cytokinesis takes place by cell plate formation, and in case of animal cell, it takes place by constriction of plasma membrane. So there are only two differences, but these are very, very significant differences. Now, two important things. When we are talking of mitosis, we need to know two types of substances. One, which stimulate that division, and the other which inhibit that division. So first category is of mitogens. Mito is for mitosis and gen is for generating. So any substance which stimulates this cell division is called a mitogen. Auxins, cytokinins, these are plant hormones. We call them phytohormones. Like in our body we have growth hormone, similarly plants also have same type of hormones. So auxins, cytokinin, they promote cell division. So whenever we are talking of cell division, the most common division that we are referring to is mitosis. So auxins, cytokinins, even steroids and insulin, these are substances which are going to promote the cell division. The cell is going to divide whenever these substances are present. And what is the process known as? The process of inducing mitosis is called mitogenesis. Again, gen is generation and genesis is the process. So cell division taking process is called mitogenesis. Then the next category is of mitotic poisons. And as the term has poison in it, it easily tells us that this is going to inhibit or stop mitotic division. That inhibit cell division, they are known as mitotic poisons. Out of many mitotic poisons, there are few very important on which the questions are asked. So now there are 2-3 more important poisons which we are going to talk about, and then we will discuss one in detail. Azide, I don't know whether you've heard of this or not, but I'm sure you've heard of this, cyanide. It is a poison. But when we say cyanide kills, it affects two different things. One, it affects the respiratory enzyme, because of which the death is instant. But the same poison also inhibits prophase. And if there is no prophase, or if the cell division stops in prophase, there would not be the further division or further stages. If no metaphase, anaphase, telophase, nucleus is not going to divide. And obviously if nucleus doesn't divide, then cytoplasmic division doesn't make any sense. So these two, they stop the cell division, they are poisons, but very specific, they stop the cell division in prophase. The next one is the most important one. It is a poison, but we are going to use it for our benefit also. It inhibits spindle formation. What exactly is this spindle formation? We talked about it in the previous lecture that spindle is just the arrangement of the spindle fibers. One end is narrow, wider in the middle, and other end narrow. This is what is called the spindle. So it inhibits spindle formation. So what happens is spindle fibers are formed but they break. So that complete assembly is not formed. And so it stops mitosis in metaphase. Two important things. Azide and cyanide inhibit in prophase. And if you're talking about colchicine, which is the most important one, it inhibits mitosis in metaphase. This is important because questions are invariably asked on this. Let us talk about colchicine first and then we'll come to one more poison. Colchicine, it is not going to let the spindles form. So if in a cell the proper spindle formation doesn't take place, so what will happen is, suppose there are 4 chromosomes, or say, why we are taking 4 is because the division that we studied with 4 chromosomes. Do you remember that these 4 chromosomes, they have sister chromatids? That means they have double the genetic material. So if DNA or genetic material is double, chromosomes are basically double the number. Ultimately they are going to split into double the number. So if you say 2n, we called it diploid. If the chromosomes split, after splitting what will happen is, there would be eight chromosomes. 4 have undergone splitting, so there are 8. But as the spindles are not formed, these chromosomes will not be properly moving to the opposite poles. So in a cell now, here the chromosome number was 4, which is like 2n in our cell's case. Here the chromosome number is 8, which is 4n. Don't you think the ploidy is changing? So the cells become tetraploid or double. So here the cells become polyploid. So colchicine, if is used, it will result in formation of a cell which is polyploid. And you know, polyploidy is a very important step in plant hybridization. The wheat that we are using or consuming these days is hexaploid. Hexaploid means 6n. So how was the 6n obtained? The normal cell is 2n, and then it was treated with colchicine so that the chromosome splits, but it doesn't move to opposite pole. If it doesn't move to opposite pole, no new nucleus will be formed, no telophase. So in one cell, the chromosome number is going to double or multiple of that. So this is how we get polyploids. And polyploids have high productivity. So we are going to use these polyploids in obtaining high yielding varieties of plant, so that the requirement of more and more food is going to be supported. Population is increasing, demand for food is increasing, but the land which is available for agriculture is decreasing, because as human population increases, you need to provide more and more land for their habitation. So how do we produce more from the less land? the most common option that comes to us is grow high yielding varieties, so that in that small land each plant is able to produce more seeds or more grains, and that can be achieved by this polyploidy. And the substance that we use is colchicine, most common substance. One more substance which acts as a poison is carbon monoxide. Actually, carbon monoxide is a toxic gas, not only for plants but even for us. It inhibits Krebs cycle. It is not affecting the complete cell division directly, but Krebs cycle is the step of cellular respiration, and this is the step which is very essential for ATP synthesis. So indirectly, carbon monoxide is going to affect cell division. How will it affect? If Krebs cycle is inhibited, ATP production will be less. If there is no ATP, then how will the chromosomes move from the equatorial plane to the poles? And if sufficient ATP is not synthesized, the checkpoints will not be activated. So indirectly, even carbon monoxide is going to affect cell division. So these are some poisons which affect cell divisions, and they are called mitotic poisons. Okay, this can be skipped. All right, now we are on another division which is very special. Let me check. There are few messages. It says, how are you live on 2 channels simultaneously? I will be live on 2 channels. One is this one, that is Let's Crack NEET UG English, and at the same time after this I'll have one more session on Let's Crack NEET UG. So there are 2 sessions which I'm going to be taking. And audio clear, hello ma'am, everything is good so far, no questions. Now what you should be doing is, when we study a particular thing, after the lesson is over, after the lecture is over, do revise that thing, and when we have lectures you can send your questions or doubts, so that maybe in the middle of the session or towards the end of the session, if there are some questions, probably I'll be able to answer those doubts if you have. So what we'll do is, I'll start with meiosis. We'll have a brief introductory part of meiosis, and then we'll start with first phase. And towards the end, maybe for 5 minutes, I'll again wait for your questions if you have any kind of doubt. And if there's no doubt, then maybe we'll proceed with what we are doing. So this is the next division. So now in cell cycle, when we are talking of M phase, that is division phase, there were 3 divisions: amitosis, mitosis and meiosis. So we are on the 3rd one. And this division is the most complicated division. And the complicated part is only one. Why is it complicated? Because there are many changes which take place only in one phase, that is Prophase I. But we'll try to understand it, we'll try to make it very simple so that you understand it in one go itself. So meiosis, the 3rd and the most special type of division. It is known as reductional division. Mitosis was called equational division. Meiosis is known as reductional division. If you're talking about mitosis, then if this is the parent cell having 46 chromosomes, it is going to give rise to 2 daughter cells, each having 46 and 46. And that is why we called it equational division. If you're talking about meiosis, it always starts with a diploid cell. So this cell is also diploid. But the cell is going to divide 2 times. in one division, which is called meiosis I, we will get two daughter cells. And these daughter cells are going to get one set. So if you're talking about humans, if this is 46 chromosomes, here is going to be 23 and 23, and these cells will divide again. The division will be called meiosis II, and we get two daughter cells from here and two daughter cells from here. Each one is going to have only one set of chromosomes, 23, 23, 23 and 23. And normally these haploid cells are the gametes. So in meiosis, there are 4 daughter cells, and in case of mitosis, there were only 2 daughter cells. These points would help us understand the difference between the two divisions. So if we start with a diploid cell, in the first meiotic division, we are getting 2 haploid cells. So reductional, that means this meiosis I is a reductional division. The 2 daughter cells having one set of chromosomes, 23 chromosomes, when they divide, they give rise to cells having same number of chromosomes. So don't you think meiosis II is like mitosis? So this meiosis I is the reductional one. This is reductional division, and meiosis II is similar to mitosis. But there are differences, it's not exactly same. But if the number of chromosome remains the same from the cell where it is starting from, so for this division, this becomes the parent cell and these are the daughter cells. So if the chromosome number is same, it is equational. So in meiosis, there are going to be 2 divisions. It is a special type of division where DNA replicates only once and the cell divides twice. This is the most important part, and that is why we need to understand this particular division very, very clearly. It starts or it occurs in special cells which are called meiocytes, and meiocytes are always diploid. Mitosis can take place in diploid cells as well as haploid cells, but meiosis will always take place in diploid cells. It involves 2 divisions, meiosis I and meiosis II. So this is just a basic information. Now let us have an overview. We saw that meiosis involves 2 divisions. So we will write those divisions as meiosis I, and I'm going to write it here, meiosis II. Okay, meiosis I is completed in 2 steps. Karyokinesis and cytokinesis. Meiosis II is also completed in 2 steps. The first step is karyokinesis and cytokinesis. Now, if a question is asked about karyokinesis, so how will you know that we are talking about, or the examiner is trying to ask a question from karyokinesis of mitosis, karyokinesis of meiosis I, or karyokinesis of meiosis II? So to make our life simple, what we will do is, we put a number. It is a part of meiosis I, so it will be called karyokinesis I. This will be called cytokinesis I, karyokinesis II, and cytokinesis 2. Now this is easy. If it is written as karyokinesis, we are talking of mitosis. If it is karyokinesis 1, it is of meiosis 1, karyokinesis 2, meiosis 2. Simple. Karyokinesis 1 is completed in the same 4 phases. What are those 4 phases? Prophase, metaphase, anaphase, and telophase. All these phases are same names. Again, to make it easy for us, prophase 1, metaphase 1, anaphase 1, and telophase 1. Karyokinesis 2 is also completed in the same 4 phases. So here we will write prophase 2, then metaphase 2, anaphase 2, and telophase 2. So again, terms are same, the phases have the same names, but how do we differentiate? If I am talking about prophase, it is of mitosis. Prophase 1, meiosis 1, prophase 2, meiosis 2. So there is no scope of confusion. Now comes the most important thing. Prophase 1 is the longest phase, and in this phase there are further phases. That means prophase 1 is divided again into phases. Let us write down these names. Leptotene. Now this term, this phase, or this name of the phase is coming for the first time. It was not there in mitosis. So we don't have to write 1, 2 or anything. It is just coming for the first time. So leptotene, zygotene, pectine, diplotene, and dikinesis. These names are coming for the first time. So this is meiosis 1 and meiosis 2. But one cell which enters meiosis 1, the same cell has to go through meiosis 2 for the complete division to take place. The cell which enters meiosis 1 comes here after completing interphase. So the cell which has done interphase is the cell which is going to enter into meiosis 1. We are talking about cell cycle basically. So in cell cycle, after interphase, when the cell divides, it can divide by amitosis, mitosis or meiosis. So cell has to do interphase. Okay, what has happened in interphase? G1, S and G2. In S phase, DNA replication has taken place. So in interphase, DNA replication has taken place, and the preparation was also there. In G1, the cell prepared for S. In S, it replicates its DNA. In G2, it prepares for division. Now in mitosis, there was just one division. But here, after G2 or in G2, whatever preparation the cell has made, all those things will be used up in meiosis 1. And after cytokinesis, when the cell has to divide one more time, don't you think it requires some raw material, ATP, some preparation? So, between cytokinesis 1 and karyokinesis 2, there is a short preparatory phase, and this is known as called interkinesis. Okay, a quick recap. What happens in cell cycle? Interphase, then division. Here we are talking about a special type of division, which is meiosis. So cell goes into G1, prepares for S phase. In S, DNA replicates. In G2, the cell prepares for division. So in case of meiosis, Meiosis I is that first division which has taken place, and all the requirements, all the preparations have been used up here. Now the cells have to divide again because meiosis is completed in 2 steps. So between first division, that is meiosis I, and second division, that is meiosis II, the cells require some preparatory time. They need to prepare few things like ATP, protein, some raw material. So all those things will be prepared in this interkinesis. How is interkinesis different from interphase? In interphase also preparation was there, but here there is no S phase. What does this mean? If you say that here there is no S phase, that means only protein ATP will be synthesized, no DNA replication. No S phase, no synthetic phase, no DNA replication. So cell is dividing 2 times and DNA replication takes place only once. Okay, let us come back to this point. And here we said DNA replicates only once and cell divides twice. This is what we were talking about. So now this is just the overview of meiosis. Prophase I is the longest phase, and in prophase I there are 5 subphases. The sequence is the same. You cannot change the sequence. It is going to be leptotin first, followed by zygotine, then pectine, then diplotin and dikinesis. This is how the changes are going to take place. So sometimes very simple questions are also asked. They give you 4 options with the same 5 names, but they are jumbled up. And you have to pick the option which has all these 5 names or stages given in the correct order. So leptotin, zygotine, pectine, diplotin and dikinesis. Once we do these stages in detail, it will be easier for you to remember the stages, because these stages, the names which are given, are because of some reason. So once we know the reason, you will automatically remember these names. So this is actually the overview of meiosis. So one cell, which has to be a diploid cell, it is going to divide by meiosis. And meiosis is completed in 2 parts, meiosis I and meiosis II. When the cell enters meiosis I, it has already completed its interphase, so it is ready with the raw material, it has double DNA. But when the cells move from meiosis I to meiosis II, they have a short preparatory phase where they prepare ATP, proteins, raw material, but there is no S phase. That means DNA replication is not going to take place. So DNA divides only once, but the cell divides 2 times. So is this part clear? Now we can come to, okay, Adarsh, I have, ma'am, okay, there is one question. Let me take this question. It says, it is given by late study. Ma'am, is that compulsory that cytokinesis should be there between meiosis I and meiosis II? Not necessary. But when we talk of theoretical part, we have to write down that stage. But if the cell anyways is going to undergo one more division, then why waste energy for cytokinesis? So these are the mechanisms which are adapted by various cells to conserve energy. So it is not compulsory. Sometimes after telophase 1, immediately karyokinesis 2 will start. Adarsh is saying not understanding, not understanding, not understanding. What is that thing which you are not understanding? So you have to give me a very specific thing. In the beginning itself, I told you that meiosis I is not an easy process to understand. It is going to take time. So that is why I have divided it in 2 parts. We are doing a basic part today, and in the next lecture, we will revise this basic part so that you also get time to read it once. And then we will continue with the next stages. I have not even started the stages. So Adarsh, you need to tell me what is that thing which you are not actually getting. Okay? All others are able to understand. This is just an overview. Okay? We are going to start all those stages one by one, and we will go a little slow because for the same reason. Others say cytokinesis. What is the problem in cytokinesis? Cytoplasm divides. Okay. Now, if you're talking about cytokinesis in plant cells have a rigid cell wall. So plasma membrane cannot move or fold. So how do you divide? You make something in the middle of the cell, and that structure grows on all the sides like a plate. So when it grows, it divides the cytoplasm into 2 parts. And if you're talking about animal cell, they have only plasma membrane, there is no cell wall. So this plasma membrane becomes folded, and those folds get deeper, 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 and finally those membranes join, separating the 2 cells. Very simple. In plant cell, it starts in the middle, grows towards periphery. In animal cells, it starts from the periphery and grows towards the center point. Very, very simple. So I would suggest that the diagrams which I made in the beginning when we were talking about cytokinesis, so go over those diagrams, and I am sure it will be clear to you. Okay, I think again the cell plate thing is there. I'll explain the cell plate thing one more time. But before that, let me just give you this brief thing, or what we can do is, we can talk about this question which you're asking here is, that is cytokinesis. Okay, this is not about the prophase I, we are talking about the doubt that you have. Suppose this is a plant cell. The 2 nuclei are already formed because telophase has completed. Now in the middle, there would be pieces of Golgi and ER which are going to migrate. What do? They secrete a material which is a pectic material, and this material will make a layer like this. This layer is called middle lamella. So middle lamella is formed, and this layer will grow towards all the sides. You have to imagine a 3D cell. In the middle, there is a small plate formed, and this plate is going to grow on all the sides. So ultimately it is going to touch the plasma membrane on all the sides. This is a section, so we have cut the cell vertically. Now both the cells, they have their cytoplasm. So the cytoplasm of this cell and cytoplasm of this cell, they start secreting cellulose. So when they produce cellulose, the cellulose will come and get deposited here from one side, and cellulose will come and get deposited here, and this is going to grow. This touches the plasma membrane on all the sides. So the cytoplasm has divided, and the structure which is formed is nothing but the cell wall. Cell wall has middle lamella in the middle, and on either side there is cellulose. And if you're talking about the animal cell, then that is very simple. This is the cell. The two nuclei are formed, plasma membrane is flexible, and so it becomes folded like this. These folds are going to get deeper and deeper, and ultimately this membrane joins here, this membrane joins here, and you get two daughter cells. Okay, so I hope this is clear now. When will 12th standard start? Very soon, very soon. Okay, got it? Aditya? Okay, you got it. I'm happy you've understood. Okay, so far so good. So I hope the doubt which you had in cytokinesis is clear now. You go over this one more time, and if you still find it difficult, maybe in the next lecture, don't forget to send or message me your question. I'll take it up again. Okay? Alright, so let us come back to the division which we are talking about. We were talking about Prophase I. Now, in the previous part, we had an overview. Now, this time, our focus is only on Prophase I. So, when we talk of Prophase I, only Prophase I, we said there are 5 sub-phases. First, leptotene, it is also known as leptonema. Leptos, because in biology, students normally find that why the words are so difficult. Why can't the words be very simple, like our simple English? The reason is the words, maximum terminology is either picked from Latin or Greek languages. This we know. These two are dead languages, they are no longer in use, we don't use them, people normally don't talk in these languages, and that is why we are not aware of these words. But if you know the meaning, then it is very easy. Leptos means thin, and nema word is always for a thread. So what is going to be this stage? It will be a thin thread stage. This is the stage where the chromatin fibers are going to be thin thread-like. Then let us come to the next one, that is our next stage is zygotene, or it is also known as zygonema. Zygo word is used when we talk of fusion of gametes. Zygote, sperms and egg, male gamete, female gamete, when they fuse, fertilization takes place and zygote is formed. So when we are talking of zygote or zygote, that means there is pairing of one male and one female chromosome, or there is pairing of chromosome out of which one comes from the male parent and other comes from the female parent. So what will happen here is pairing of chromosomes, and this pair is called homologous pair. So now once you understand the meaning, then the words don't sound that difficult. The next, pectine. Fourth is diplotin, and fifth is dikinesis. Okay, so as I gave you the specific things for leptotin and zygotin, I will tell you about all three stages. The reason why I'm not telling you right now, because this will be easier by this name when we actually draw that stage and we try to understand what is the change which is actually taking place. Okay, so 5 subphases of prophase: leptotin, zygotin, pectine, diplotin and dikinesis. This sequence is fixed, we cannot change it. So these are the 5 subphases of prophase I. Actually, I was planning to give you some idea about leptotin and zygotin today itself, but because there were questions and I thought that probably there is maybe you might need little more time to recap this. What I'm going to do is I'll start with the same stages, leptotin, zygotin, in the next lecture. Okay, I wanted to give you some idea. So in leptotin you would find a thin thread stage. Very brief is what I'm going to tell you. Suppose this is a cell, and don't worry if you don't understand it in one go, and this is the large nucleus. In leptotin stage, the cell has entered prophase I. In leptotin stage, the threads are thin, and all these threads, they are very thin and they are attached to the nuclear membrane on one side. One end is free, other end is attached to nuclear membrane. They are thin, but condensation process starts. And condensation is not everywhere. If you remember, when we talked of chromatin fiber, we said it condenses and you start seeing the chromosome-like structure, right? Here we showed the complete condensation, but actually condensation takes place at intervals. So don't you think these threads are appearing beaded? Like you have beads in a thread? And when all these beaded fibers are attached at one point and free at the other point, it also gives an appearance of a bouquet. So this leptotin stage is also known as bouquet stage. So what has happened? Chromatin fibers have started to condense. Condensation is at intervals, so fibers have a beaded appearance. And all the fibers, they are attached to a small part of nuclear membrane from their one end. So one end is attached here and the other ends are free. And that is why there is a bouquet-like appearance. Okay, just a brief thing. In zygotin, what will happen is the pairing will take place. Nuclear membrane will still remain intact. The chromatin fibers which were beaded attached to the nuclear membrane, they detach from the nuclear membrane. And now you see the pairing. So this is one pair. We started with 4 chromosomes. So, or 4 chromatin fibers. So this is one pair and this is one pair. Why we call this a pair? Because this chromosome was received from the father and this was received from the mother. Such a pair is known as homologous pair. Just introduced few terms. We will take up these terms in detail, all the stages. So in the next lecture, we will be starting with Prophase I, same 5 phases in detail. But go over these terms so that when we take up in the next lecture, you have some basic idea of this. So this is what I had planned for this lecture. Don't forget to subscribe, and whenever you decide to go for the subscription plan, whichever plan you finalize, monthly or 3-month plan or an yearly plan, don't forget to use this code so that you are eligible for that discount. I'll see you in the next lecture. Till then, take care, revise everything properly and come prepared. See you then. Bye-bye.